All right, hey guys, welcome to another part 2 of my series for tomorrow's exam, June 18th, 2019, Living Environment Regents Study Guide. I pulled up an old 2010 exam only because I really like this exam. And what I want to concentrate today is part D. So I'm going to take you to part D. Do you remember what part D is? It's the lab section, the 4 mandated labs. Now, let me see if I can get a pen to help me navigate through this. All right, guys, I found a pen. Found the pen. I'm back to my laptop. Better sound. Still don't have my digital pen, but it's okay. So let's start with part D. Okay, part D is oh, this is interesting. Oh, this is supposed to be a check. Okay, part D is about the 4 mandated Regents labs that you did in class. If you didn't take those, if you didn't do one of those 4 labs and you're allowed to take the exam, one, the school's breaking the rule, a state rule, that because you're supposed to complete the 4 mandated labs in order for you to take this Regents. So this section is about the 4 labs. The 4 labs are diffusion through a membrane, relationships and biodiversity, Beaks of Finch, and diffusion through a membrane. I think I said that already. So, all right. So let's go ahead and get started. Where's my pen again? Here it is. Okay, so for number 65, I'm going to do exactly what I did in my last video, that I just read the questions and I went ahead and broke down the answers. So for number 65, the amino acid sequences of 3 species shown below were determined in an investigation of evolutionary relationships. And if you remember in class, we did this particular question with relationships and biodiversity. Particularly, we were discussing Botanicurus, species X, Y, and Z, and we had 7 tests to do. And this one was particular with protein synthesis, and you have to compare their amino acids. So let's see what this question wants. But remember that you have to start from the top and try to figure out how many questions does this pertain to? So let's read the directions. It says 65 to 77. For those questions that are followed by 4 choices, circle the number of choice that of those given best completes the statement or answer the question. For all other questions in this part, follow the directions given in the question and record your answers in the space below. I want you to remember that this is done in pen. All short responses are done in pen unless they ask you to draw or it's a graph. Okay, based on these data, which 2 species are most likely related? Support your answer. So maybe I can change my pen here. So we have to figure out if species A, species B, species C, which ones are closely related to each other. Is A closer to B? Is A closer to C? Is C closer to B? Etc., etc. We have to compare their amino acid sequence. Here, all 3 of them are the same. Here, all 3 of them are the same. Here, A and B are similar, and species C, so we can already see that A and B are starting to, look alike. But wait a minute, here A and B have a similarity. Here, they're all similar. Uh-oh. Here, A and B is similar, and here they are all similar. So if we were to count how many times A and B are similar, it's a lot more, well, 2 times more than species Z. So let's compare A and B together. They have 1, 2, 3, 4, 5, 6. Wait, let me start that over. They have 1, 2, 3, 4, 5, 6 things in common. So they have 6 things in common. Ay, this is not looking good. Let's see how many B and C have in common. 1, 2, 3, 4 things in common. Let's see how much A and C have in common. 1, 2, nope. 3, 4, nope. 5. So the winner is species A and species B. So check it out. It also says support your answer. It says support your answer. So you can't just put species A and B. You have to say why. So what you can say is species A and B are the 2 species most closely related to each other because they have the highest number of amino acid sequence of and just in case you're like, what? Amino acid sequence is in the question. They have 6 similarities, 6 amino acid sequences. And that's it. You do not have to reinstate the question at all. You don't have to reinstate the question. Just go straight forward to the answer. Okay, next. Let me go back there. Next, 66. A student carried out a lab activity where she was asked to squeeze a clothespin as many times as she could in 1 minute and record the number. Remember, this is when your teacher gave you a clothespin and you had to squeeze it for a minute and your hand cramped up. This was particularly talking about muscle fatigue, the muscular system, the respiratory system, the circulatory system, the respiratory system, you're breathing in oxygen and all this oxygen being rushed to your muscle cells that are in need of oxygen for ATP production. She immediately tried the same activity again, thinking she would do better the second time, but the number was lower. She immediately tried again, but the number was lower still. State one reason why she continued to get lower numbers, even though she tried to increase the number of squeeze several times. So, what do you think happened here? I'm going to give you some time to think. So, if you remember in this experiment, it was about muscle fatigue. So, what happens when you work out a lot? What happens when you work out a muscle over and over again? You get muscle fatigue. So the answer could be, one reason is that she developed muscle fatigue. Another reason, muscle fatigue is caused by the buildup of waste. That waste is called lactic acid. Then it makes your muscles sore, but then your body gets rid of it. You could have also written waste built up. You could have also written ATP was used up, because you know that when you're using, when you're exercising, your muscles are working, you are using ATP, and that's also creating a waste. Let's go to the next question. The next question is 67. So remember this relationships in biodiversity? This relationships in biodiversity, no, this one's diffusion through a membrane. This is when you made a cell and when you took a plastic tubing, that probably felt and looked like a Ziploc bag, and you tied the ends and you put 2 things in here, 2 solutions. You put glucose and you put starch. Close it up, put it in a beaker of water. Then you put in the water iodine, which is a starch indicator. So let's see what they want us to find out. Identify the process that would most likely be responsible for the movement of glucose from inside the artificial cell to the solution outside of the cell. This bag is the artificial cell membrane. This bag is supposed to represent a cell, an artificial cell, particularly just the cell membrane, because we're looking at things, molecules moving in or out. Only diffusion though, because this is not a real cell, so the only thing it could really do is diffusion, movement of molecules across the cell membrane from high concentration to low concentration. Identify the process, diffusion, or passive transport. Diffusion is the answer, or passive transport. You do not get credit for saying anything else. Diffusion or active transport. Next, 68. State why the evolutionary relationships shown in diagram 3 are not supported by the data provided by the stem cross sections in diagram 1. Okay, so let's take a look here. Notice how it says numbers 68 through 71 on the information below. So this pertains to 1, 2, 3, 4 questions. Okay, each worth 1 point. This can make or break you getting from a 60 to a 65, or a 63, which is still failing. Remember, a passing is a 65, and you need 41 points to pass. So make sure you answer every question, read the directions, take your time, the most important 3 hours of the year. You worked all year for this, so let's go. Scientists attempt to determine the evolutionary relationship between 3 different plant species, A, B, and C. In our case in class, it was Botanicurus species X, Y, and Z. In order to do this, they examined the stems and DNA of these species. So here we have a cross-section of the stem, and here we have electrophoresis, and here we have the phylogenetic trees. Diagram 1 represents a microscopic view of the cross-sections of stems. Okay, no problem. Stems of these 3 species. DNA, let me switch this up. DNA was extracted from all 3 species and analyzed using gel electrophoresis. That's this over here. That is gel electrophoresis. The results are shown in diagram 2. So here, diagram 2 is the results of electrophoresis and analyzed using gel electrophoresis. The results are, okay, based on the data they collected, they drew diagram 3 to represent the possible evolutionary relationships. Let's take a look at number 68. State why the evolutionary relationship shown in diagram 3 are not supported by the data provided by the stem cross-sections in diagram 1. Okay, so let's take a look at diagram 1. Do all 3 stems look similar or different? To me, they all 3 of them look different. And let's see what it says here. In electrophoresis, it says that species A and B, wait, let's see. This shows that C has only 1 similarity. So this could be a little tricky to analyze based off of this question, so you have to be very careful. So what you could say is, according to diagram 3, C should look different from A and B. So according to this, C is not closely related to A and B. A and B are closely related. So if that's the case, the stem of this should be different. So here, what you can put is, according to diagram 3, C should look different from A and B. That's it. That's all you have to say. Don't reinstate the question. They only give you 2 measly lines. Moving on. Explain how the DNA banding pattern in diagram 2, okay, supports the evolutionary relationship between the species shown in diagram 3. So let me just undo this. So they're saying that A and B are closely related and C is not. Let's take a look. You want to move across, looking across to see which bands are similar. So when we look across here, A and B check out. This one doesn't check out. This one's like by itself. Oops. Here, A and B check out. This one is by itself. This one's by itself. Let's see. Let's see if this one. So what you can do is take scrap paper, put it over this. Let's see if this can do the, ooh, it did it. So you want to take a scrap paper and you want to move it. You want to move it down and see. Now let me actually, this is worth the time. Hold on. This is definitely worth the explanation. Okay, so I'm going to take my little fake scrap paper and I'm going to work my way down. Boom, A and B similar. C is by itself. Boom, A and B similar. This one's not aligned, so that's not, C is not coordinating well here. Okay, B is by itself, that's fine. A and C are similar here. A and C are similar. So let's see, let's count. You have to count. So let's see how many times A and B is similar. 1, 2, and 3 down here. I'm only looking at rows A and rows B. Let's see how many times A and C are similar. 1 and 2. Let's see how many times B and C are similar. 1. So, explain how the DNA banding pattern in diagram 2 supports the evolutionary relationship between the species. Diagram 3, right here. What you can say is A and B have the most bands in common. And that's it. A and B have the most bands. These are called bands. Band, 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 band. Next. This technique used to analyze DNA involves the, so before we even answer that, you have to know what electrophoresis is. Electrophoresis is a machine used to compare DNA from different species. You can find out what animals are related to each other, who's the mom. Let's say you have a litter of puppies and you don't know if all those puppies belong to a particular mom dog. So you take the DNA of all the puppies, you take the DNA of the mom, for each well here, you could put mom here and 2 puppies. Run this machine. It's going to run from negative to positive. You turn on the machine, electricity flows through the machine. The DNA is going to go from negative to positive, so it's going to move this way. Okay? It's going to move this way. And then enzymes are going to cut the DNA strands that was put here. So you put DNA in here. And enzymes cut in special places. And in lab, you remember when you were cutting between C, C, G, G? So that's basically what's going on. So here we have this result showed that A and B are similar. So let's see. This technique used to analyze DNA involves the synthesis of new DNA? No. We put the DNA of existing, already living, you can't just make new DNA strands. DNA comes from living things. Separation of DNA fragments in the basis of size? Yes, that's basically what it's doing. Production of genetically engineered DNA? Nope. Removal of defective genes from DNA? No, we're not doing that. This is a test to compare DNA for analysis of relationships. Next, 71. Explain why information obtained through DNA analysis is more reliable indicator of evolutionary relationships than observations of stem cross sections with the microscope. So they're saying basically, why is this more reliable than this? Why is looking at DNA more reliable than looking at a stem of a plant? And that's because DNA analysis is more reliable since the more similar the DNA, the closer the relation. The closer, let me move this. Let me try to move this now. Technology, guys. Technology. Do this. The closer. Okay, let's start this over. Okay. DNA analysis is more reliable since the more similar the DNA, the closer the relationship. Don't write in the space. Just keep going under. It's fine. Okay? We know from scientific research that DNA is the most reliable source of knowing when something, a species, an organism, a living thing, is closely related to each other. Okay, moving on. Easy peasy here. Remember, start from the top. Start from the top. Base your answer to questions 72 to 74. So all of these are in common except for 75. On the information below and on your knowledge of biology. A wet mount slide of red onion cells is studied using a compound light microscope. A drawing of one of the cells as seen under high power is shown below. So here you have a slide. You have onion cells under it, and then you have a cover slip. There's definitely water in here as well. If you take one of these cells and you magnify it, you get this beautiful cell. This is one cell. I want to emphasize that this circle around is the microscope eyepiece view. It's the microscope field of view. This is not another cell. This is not another cell. This is not another cell. This is the field of view. When you put your eyeball on the microscope to view it, this is the field view, what you see. This is the cell, the specimen. Let's answer 72. On the diagram below, label the location of each of the cell structures. So this will never change. Let's see if I can get the, okay. This one right here, this outermost layer of a onion cell, what are onion cells? They're plants. So this right here is the cell wall. This right here is the cytoplasm. And this right here, the layer that comes second, outermost cell wall, inner layer, cell membrane. This you write in pen. It didn't say draw or anything. Okay? Okay, just checking my time. 73, describe the proper way to add a salt solution to the cells without removing the cover slip. So this is the case of you have the slide and the cover slip. If you remove the cover slip, you're going to ruin the whole specimen. What you do is you put a piece of tissue paper on one side and the medicine dropper with the salt water solution on the other, and the tissue is going to begin a flow of movement. So the water that was there will go to the tissue, giving room for the salt water to come in from the other side. But how do you explain this? So what you say is the following. Place salt water solution on one side of the cover slip, period. Then draw the salt water solution under the cover slip by placing a piece of paper towel on the opposite side. And that's it. Place saltwater solution on one side of the cover slip, then draw the saltwater solution under the cover slip by placing a piece of paper towel on the opposite side. Okay, that's it. All right, next. In the space below, sketch how the cell would look after the saltwater solution is added to it. So all you have to do is draw this over here, but you see the shaded area? The outermost layer of the shaded area, again, is the cell membrane, and the cytoplasm is the jelly-like substance in between. You just have to keep the cell wall the same exact way. So let me see if my pen is going to, and let's give it the real onion color. Let's see if it can help me out here. Okay, yeah. The only problem is I can't, no. Let's see if I could change that. Okay. Let's see. So you're going to draw the cell membrane, right? I'm sorry, the cell wall. And then you are going to draw a very small shrunken cell membrane and cytoplasm. And that one you should definitely fill in. So it's the cell wall stays the same. I know the shape isn't exact, but hey, this will do. This will get you the point. And then the cell membrane, so the cell wall stayed intact, and the cell membrane and cytoplasm shrank, because when you put salt water in, the salt diffuses in from high concentration to low concentration, and the water that was in here comes out. Okay, moving on. 75. A student added an enzyme to a test tube containing a sample of DNA. After a period of time, analysis of the DNA sample indicated it was now broken into 3 segments. The purpose of the enzyme was most likely to cut the DNA at specific locations. Oh, definitely, because that's what happened to us at CCGG. So when we were doing the electrophoresis. So 1 is definitely the answer already. Move the DNA to a different organism? No, it's in a test tube. Why would it be in a different organism? So it's not this one. Copy the DNA for a protein synthesis. We weren't copying, we were just cutting them in segments. Alter the DNA sequence. We did not change the nitrogenous bases, ATCG, so we were not manipulating with that. So the best answer is 1. Let's keep going. Oh, the Beaks of Finch. Remember when you were competing for seeds? That was pretty fun. Okay, so here we go. Before we answer the question, remember that This was called the Beaks of Finch. And this is about Charles Darwin, natural selection, evolution, how Darwin saw the same species, but in different parts of the island, they had different beaks that pertained to their niche being their role in their environment, and their beaks. Their beaks depended on what they ate. Were they carnivores? Were they herbivores? Etc., etc. The 4 different types of beaks shown are most likely the result of gene manipulation, natural selection, unchanging environmental conditions. We already know that's wrong because their environments were different, so that's why they have different beaks. Patterns of behavior learned from parents, not relevant. So for number 76, it's natural selection. Natural selection. Natural selection, survival of the fittest. Their beaks are special to the type of food they eat, because if they don't have the special beaks that help them eat, they will not be able to survive. They need the right tool, the right beak for the food that they have to eat. It's not gene manipulation. So the answer is natural selection. Next. Scientists observed that when 2 closely related species of predator birds live in different areas, they seek prey early in the morning. However, when their territories overlap, one species hunts at night and other hunts in the morning. When these 2 species live in the same area, they apparently modify their, what do you think they modify? Their niche. They modify their niche. They modify their role. Niche is the role that an animal plays in its environment. Now, I never seen a test being only 77 questions long, but that's it. That's it for this test. Now, we didn't bump into certain questions that I wanted to get to, but again, the living environment test is very random. It's very random. So it didn't do protein synthesis. See, this is protein synthesis, but in other tests, they had you do the messenger RNA and then go look at the chart and then get the amino acids, which I know you would be able to do. All right, guys, again, look out for my videos, and I'll be posting a couple of more, so stay tuned.